Hello and welcome back. Today's mini YouTube lecture is a review, actually, and it's going to be a review of the structure of DNA. And today we're going to be talking in particular about why we keep on saying 5' to 3', 5' to 3', and why is that so important in discussing the structure of DNA. All right, so let's take a look at these two nucleotides. All right. Now, you'll recall that in one of our last lectures, we were talking about how you can take two nucleotides and they can undergo a dehydration reaction, right? That we form a new bond between two of these monomers. And in this example right here, we've got an adenine and a thymine that have formed this new bond between them. And that bond is called the phosphodiester bond. So let's write that out. Phospho, right? Oh, sorry about the terrible handwriting. Diester bond. All right. And you can see that I've drawn it there as this phosphate that connects this ribose sugar to this ribose sugar, right? Now let's cover this up real quick and let's take a look at the single nucleotide down here and all of the features that make it a nucleotide. Right? You'll remember that a deoxyribonucleotide is a 5-carbon sugar. So we've got the 1', 2', 3', 4', and 5' carbons. And then there are two very special functional groups that are hanging out off of this 3' carbon here that I've labeled and the 5' carbon. All right? So if you remember your organic chemistry, off of the 3' carbon is the 3' hydroxyl. So I'm going to write that down right here. 3' hydroxyl. Right? That's that OH right there. And off of the 5' carbon is the 5' phosphate. So 5' phosphate. Now these are the two functional groups, the hydroxyl and the phosphate, that participate in the reaction that joins two nucleotides together. That is the dehydration reaction. So you can see right here where we've got this dinucleotide, and here's this incoming free nucleotide right here. And so you can see where the phosphate meets up with the hydroxyl, and then we can make water go away, right? Causing a dehydration, and that makes this strand of DNA one more nucleotide longer. And so if we want to make this even more long, like add another nucleotide, we add on another nucleotide to that 3' hydroxyl, and so on and so forth, elongating the strand of DNA. All right? Now, you know that DNA is not single-stranded, right? Hopefully by now, you may have already had a high school DNA class where you know that DNA is double-stranded. And in cartoon form, that typically looks like this, right? Like this double helix, right? We're going to talk a little bit more about the structure of DNA later, but here are these two lines that run parallel, right, to each other and have this, if you can imagine, a three-dimensional helical structure. This is our double helix. Well, let's make this simple, okay? Let's take this and let's untwist the helix and stretch it out. So we've got a strand right here and a strand right there, right? So we've untwisted that helix. Now, I want to describe a very important structural component of DNA, all right? So here at this top strand, let's take this, let's see, the 5' phosphate. We're using orange for phosphate, right? So we've got the 5' phosphate here, and down at this end, we're going to have a 3' hydroxyl, which I've labeled before in green, right? So down here, I'm going to label it 3', okay? So this strand runs 5' in one direction, at one side, to the 3' pole, right? So there is polarity. And you can assume that this means that this up here, this is the side with the free phosphate end, and down here, this is the side with the free hydroxyl end, okay? But double-stranded DNA is anti-parallel. And you're going to hear me using that term a lot. Antiparallel means this, right? If you have the 3' hydroxyl group down at this end, on the opposite strand, the complementary strand, you're going to have the 5' phosphate. Wow, that is a terrible 5. That's even worse. Okay, I'm just making it worse here. All right, so let's go down to the other end. So if you've got 5' here, what's going to be down at this other end? The 3' end, right? So this feature of this nature of double-stranded DNA, we have a name for it. It is called antiparallel. So ds-DNA, and the little ds stands for double-stranded DNA, is antiparallel. And that has incredibly important implications for how DNA is copied by our favorite DNA copying enzyme, DNA polymerase. Okay, so let's get a little preview of that right now, shall we? I am going to draw these two strands pulling apart, but I'm going to do so in the middle of this strand right here, okay? So there's a little bubble. I'm going to call that a replication bubble because that's where replication begins, in these little bubbles, which you can actually see with an electron microscope, all right? So we're still going to keep our conventions here, 3', 3', 5', and 5', okay? And the enzymes that copy DNA, right? DNA helicase, DNA polymerase, DNA ligase, and others that we will meet soon, right? Start laying down new copies of each of these strands, all right? Based upon the DNA sequence of the template strands. Remember that word template? A template is something that you copy. So here I've drawn in black. This is our template strand. We can also call it the parental strand because it's the DNA that's being copied. And then here inside of this replication bubble, the newly formed DNA strands are the ones that I've drawn here in magenta, okay? Now, over time, this replication bubble is going to spread outwards, all right? So in the next step, I'm going to show that DNA replication is bidirectional. That means it goes both ways. So I'm going to write that down. DNA replication is bidirectional. All right? From that bubble, it spreads outward. Okay? So I'm going to draw this bubble a little bit longer. And, all right, does it make sense that now that that bubble's a little bit longer, our newly replicated DNA is also a little bit longer, right? And at the end of DNA replication, what are you going to get? You're going to get two new strands, excuse me, I should say two new copies of double-stranded DNA, right? So in black right here, that's the parental strand, right? And in magenta, that's the newly formed daughter strand. But we don't just have one copy of the DNA anymore. We have two copies, okay? So let's put right here, this black is the OG, the original parental strand. And in purple is the newly synthesized strand. Boy, my penmanship is not on point today, all right? So that is essentially what happens in DNA replication. We get a splitting apart of the strand, we get formation of a replication bubble, DNA replication is bidirectional, and we get the synthesis of two new strands at the end of DNA replication. All right, that is DNA replication in a nutshell, and in the next video, we're going to go over the nuts and bolts of what is happening inside of that replication bubble. All right, see you next time.